Remember that blood is a primary carrier of oxygen throughout the body. When you are injured while playing, you can see blood flowing outside your body. You are also familiar with blood's various components. That is, blood is made up of blood cells and plasma. Recall that red blood cells are the RBC, white blood cells WBC, and platelets are the three types of blood cells found in our bodies. Have you ever wondered what happens if you don't have enough blood cells? Individuals with this condition will thus require a blood transfusion. But my question is, can we take anyone's blood? Or do we have to check some criteria? In this video, we will be discussing the different blood groups and the RH compatibility in humans, along with the mechanism of blood clotting. First, let us check the ABO blood grouping. The human blood group is determined by the codominant alleles. Recall that an allele is one of the several forms of genetic information that is present in our DNA at a specific location on a specific chromosome. We have two ABO blood type alleles. So can you guess the reason? It is because we inherit one from our biological mother and one from our biological father. What is a genotype then? The genotype is a description of the pair of alleles in our DNA. As there are 6 different alleles, there are 6 different genotypes in the human ABO genetic locus. Do you know how the ABO blood grouping is determined? ABO blood group system is a classification of human blood based on the inherited properties of RBC or the red blood cells, that is the erythrocytes, as determined by the presence or the absence of surface antigens A and B carried on the surface of the red cells. And also, plasma contains 2 natural antibodies which are produced in response to the antigen. Before we evaluate the compatibility of blood groups, we need to know the different blood types in humans. As you can see on the screen, there are 8 different blood types and each individual has one of these 8 blood types. We can infer that the distribution of antigens and antibodies in the 4 groups of blood, that is A, B, AB and O. Blood transfusion can create major issues if not properly monitored. To avoid the serious problems with blood clotting or the destruction of red blood cells, the donor's blood must be carefully compared with the recipient's blood before the transfusion. It's time to check the compatibility of each blood group. Let us fill this table one by one. Here we can see the 4 blood groups A, B, AB and O. Blood group A has an antigen A on RBC and anti-B on the antibody in plasma. So the individuals with blood group can donate their blood to the individuals with blood group A and O. Blood group B has the antigen B on RBC and anti-A on the antibody in plasma. So the individuals with blood group can donate their blood to the individuals with blood group B and O. Again, blood group AB has antigen A and B on their RBC, but they lack antibodies. Hence, individuals with different blood groups can donate their blood to individuals with blood groups AB. But individuals in the AB blood group cannot donate blood to individuals other than the AB group. Therefore, individuals with the AB group are known as universal recipients. Finally, let us check the detailing of blood group O. Individuals with blood group O lacks antigen in RBC. The antibodies seen in the cell are anti-A and anti-B. Hence, the individual with the O blood group can donate their blood to any other individuals. What does this infer? Individuals with blood group O are universal donors. Let us now try to focus on the Rh grouping system. The genetic information of Rh factors is also inherited from biological parents. There are two different alleles for the Rh factor known as Rh positive and Rh negative. So how will we represent them? Individuals who are Rh positive have at least one Rh plus allele or Rh positive allele but could have two. Their genotype could be either Rh positive, Rh positive, Rh positive or Rh negative. Individuals with Rh negative has a genotype of Rh negative, Rh negative. But what is an Rh antigen? The Rh antigen is similar to the one present in the rhesus monkey and is also observed on the surface of the RBCs of a majority of humans. Hence, the antigen is known as the Rh antigen. An Rh negative person will form specific antibodies against these Rh antigens if exposed to the Rh positive blood. So, the Rh group should also be matched before transfusion. A rare case of Rh incompatibility has been observed between the Rh negative blood of a pregnant mother and the Rh positive blood of the fetus, resulting in erythroblastosis fetalis. Let us check this condition in detail. Consider the situation if the father's blood is Rh positive and the mother's blood is Rh negative. The fetus blood is Rh positive. During the delivery of the first child, there is a possibility of exposure of mother's blood to fetus blood that further development Rh antibodies in the mother's blood. In the subsequent pregnancy, the mother's blood can leak into the fetus blood and destroy the fetus RBC or can cause severe anemia and jaundice in the fetal baby. Can this condition be avoided? If so, how? This can be done by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. Imagine a scenario where your body gets injured and blood starts flowing out. After a few days, you will notice blood scum in that particular region. How does this happen? Which factors contribute to the clotting of blood? Let us find out. The mechanism which helps in the clotting of blood and preventing the excessive loss of the blood from the body is termed as clotting or the blood coagulation. Here comes the role of fibrins. So what are fibrins? To explain this question, we should focus on the mechanism of blood clotting and its associated terms. It is a complex series of sequential changes occurring in the injured part of the body. Platelets stick to the damaged tissues and release a phospholipid known as the platelet factors. The cells present in the injured region also secrete another factor named thromboplastin. These factors combine with calcium ions and forms an enzyme named prothrombinase. Meantime, the blood platelets disintegrate and release platelet thromboplastin. After that, in the presence of calcium ions, the prothrombinase inactivates heparin. Heparin is an anticoagulant of the blood. When heparin is inactivated, prothrombinase smoothly catalyzes the breakdown of prothrombin, an inactive plasma protein, into thrombin and some small peptide chains. Thrombin, acting as an enzyme, first breaks down the fibrinogen, that is a soluble plasma protein, into its monomers, that is the fibrin monomers, and the blood cells. Eventually, these monomers become thin, long, solid fibrin fibers form a dense network around the wound. In this network, blood corpuscles becomes intertwined. As a result, a red clot forms at the wound 2 to 8 minutes after the injury. It prevents further bleeding. Soon after, the clot begins to contract and a pale yellow fluid identified as serum begins to ooze out from it. We know that blood is transported throughout the body, aiming for different functions. But are you familiar with the differences between closed and open circulatory systems? What is meant by the cardiac cycle? Get all these questions answered in the upcoming videos. Now, let us summarize what we have learned.
Manorama Horizon. Learning made simple.